Hi everyone. In Lab 4 you learned how proteins are made, and your post-lab assignment requires you to use that information to determine the amino acid sequence of a particular protein. This can be a bit confusing, so I want to work through an example with you. If you haven't already done so, please read through Lab 4 and watch the videos that are linked in the handout. If you're ready to start the post-lab assignment, let's first work through this example. Remember that protein synthesis occurs in a couple of important steps. In the first step, we use the information in DNA to make a complementary strand of messenger RNA. This process is called transcription. The first thing we have to do, though, is figure out which of the two DNA strands is used to make mRNA. Remember that in transcription, RNA polymerase moves along DNA in the 3' to 5' direction to create the messenger RNA strand. So the strand that we'll use here to do that is this one. Remember that the messenger RNA strand that's produced is complementary to the DNA strand, much like the two DNA strands are complementary to each other. As you can see in the DNA strands, C and G are complementary or always paired together, and A and T are complementary or always paired together. There is, however, one key difference in RNA. The thymine, or T, is not found in RNA. Instead, the complementary base uracil, which is noted with the letter U, is included. So with this in mind, let's begin transcribing the DNA strand to make messenger RNA. The first base in the strand here is G, so the complementary base will be C. Moving along, you can see that the next base will also be C, and the third base will be G. If we continue along the strand, you should see that the next 3 bases included in the mRNA strand are A, U, and G. Pause the video and take a minute to finish transcribing the strand. Once finished, you should have a messenger RNA sequence that looks like this. Once again, take a minute to pause and compare your answer with the sequence shown here. The messenger RNA sequence that's produced is like one big long genetic sentence, and to understand that sentence, we have to translate it. Each of the words that makes up that sentence is called a codon, and each is 3 letters long. But before we can figure out what those words mean, we have to determine where the sentence begins. The first word is always the same, and it consists of the letters AUG. So the next thing we need to do is scan the messenger RNA sequence to find AUG. In this example, AUG is here. Once you know where the sentence begins, you can determine where each of the remaining words are that comprise the genetic sentence to begin the translation process. Now, I'm more of a visual learner, so I find it helpful to copy the messenger RNA sequence and visually separate each of the words by drawing a line between them. So pause the video and use the space provided here to do that. Once you've drawn lines between each of the words, your strand should look something like this. And now we're ready to determine the amino acid sequence. To do that, we have to use the codon dictionary, which is shown here to the screen on the right. Using the codon dictionary is pretty simple. You're going to start by locating the first letter in the word on the left side of the dictionary, move to the top to find the second base or the second letter, and then move to the opposite side there to find the third base. So we already know that the first word is AUG, and that's pretty easy to spot on the Codon Dictionary here as it's highlighted. As we can see, the Codon Dictionary tells us that AUG stands for MET, which is the amino acid methionine. So let's work through the next one together. So our next word is GAU. So we're going to first start by locating G along the left side of the dictionary. Our second letter is A, which is going to fall in this third column. And then we're going to find our third base again, which is U. So we're going to move on down here, and then we find it there. GAU stands for ASP, which we know is the amino acid aspartic acid. Pause the video again and use the Codon Dictionary to determine the remaining sequence of amino acids. Okay, the resulting sequence of amino acids should look like this. You should have also noticed that the next to last codon, UGA, does not translate into amino acid. Instead, as noted on your Codon Dictionary, UGA is a stop codon. That indicates that this is the end of the genetic sentence. So again, our stop codon in the above sequence here was UGA. All right, you should then be able to use this information to answer the last question. And I hope this helps you understand how to complete this week's post-lab assignment. If you need any additional help, please let me know. Good luck.